Hello, I am Hoài Thu Thái, a researcher at Sanofi. On behalf of my co-authors, I'm pleased to give you a presentation entitled Model-Informed Drug Development to Establish Izatuzumab-Sarclisa Combination Dosing Regimen in Relapsed Refractory Multiple Myeloma Patients. Multiple myeloma is the second most common hematological malignancy and remains incurable in elderly patients with a median survival of about 6 years. This disease is characterized by the production of cancerous plasma cells in the bone marrow, which secrete excessive amounts of abnormal antibodies called M protein, which is a major tool for response assessment in multiple myeloma. CD38 is a type 2 transmembrane glycoprotein. It was found to be widely expressed in almost myeloma cells, while its expression is low in normal cells. Izatuzumab is a CD38 monoclonal antibody with multiple modes of action for killing tumor cells through direct tumor targeting and immune cell engagement. To support the drug development of izatuzumab in multiple myeloma, several modeling and simulations were performed from preclinical to market approval, which shows a good application of model-informed drug development in oncology. This MIDD can be summarized by forming analysis. We started with the preclinical data by performing PK/PD analysis in normal and tumor-bearing mice to define the target threshold concentration for tumor eradication in order to support first-in-human study design and to guide the dose escalation. Then, when the data of the first-in-human monotherapy study became available, we conducted population PK exposure response analysis and disease modeling to support the dose selection for phase 2 monotherapy. A new dosing regimen with loading dose were proposed to izatuzumab program. Leveraging the MIDD from the monotherapy, the loading dose was implemented in the combination study. The same methodology was applied to support the combination dose selection using data from phase 1 studies and allowed to recommend izatuzumab dose for the first pivotal phase 3 ICARIA trial and also for other phase 3 studies. The most recent analyses were performed when ICARIA data were available in early 2019. Population PK exposure response analysis were conducted to support ICARIA filing. Then joint modeling of serum M protein and progression-free survival was performed to build a disease model platform, including clinical primary endpoint, and allowed to confirm the approved dose of izatuzumab. Now I will go through its analysis and its impact on izatuzumab drug development. The first work was the population PK/PD analysis in normal and tumor-bearing mice with the aim to evaluate anti-tumor activity of izatuzumab in mice and to help predicting active doses in humans. PK and tumor volume data from 2 preclinical studies with single and repeated administration were used. The Simeoni PK/PD model with tumor growth inhibition was developed and described well the tumor volume for control group as well as for the treatment groups at different dose level from 2.5 to 40 mg/kg. This model allowed to define the target threshold concentration for tumor eradication in mice. The first-in-human phase 1 study of izatuzumab started at very low dose because the MARBL approach was used. The dose escalation was divided into 2 parts. The accelerated escalation with only 1 patient per cohort, that is dose up to 0.1 mg/kg every 2 weeks, and then the basic escalation with 3 to 6 patients per cohort, testing a dose range from 0.3 to 20 mg/kg every 2 weeks. Based on the overall response rate observed in the dose escalation, 10 mg/kg every 2 weeks was selected for the expansion cohort with 18 patients. The safety data show that izatuzumab has a manageable safety profile. MTD was not reached for all tested dose up to 20 mg/kg. However, an expected lower response rate was observed for the expansion cohort at 10 mg/kg every 2 weeks with the preliminary result of March 2014. To better understand the exposure-efficacy relationship of izatuzumab and to support dose selection, the following model and simulation strategies were used. We first developed a population PK model and compare the predicted izatuzumab concentration with the threshold level for tumor eradication obtained from mice. Then exposure response analysis was performed to characterize the relationship between PK parameters, IUC or CTRAF, and the response rate. In parallel, we developed a disease model of tumor burden, i.e. serum M protein. Both approaches allow to support dose selection for izatuzumab, and we built a strong collaboration between modeling and simulation and biostatistics team. This slide presents the population PK analysis. The preliminary two-compartment PK model with a parallel linear and nonlinear elimination was developed using phase 1 data. Due to the target-mediated drug disposition in some patients, we observed lower exposure in patients from the expansion cohort compared to those in the escalation at 10 mg/kg every 2 weeks, which explained the unexpected lower response rate in the expansion. The simulations were then performed for 3 dose levels in the original phase 2 protocol, 3 or 10 mg/kg every 2 weeks, and 10 mg/kg every 2 weeks, then every 4 weeks. The simulation showed that the simulated concentration was generally below the threshold concentration for tumor eradication in mice, even for median patients. The exposure analysis and disease modeling of serum M protein were performed afterwards. CTRAF at 4 weeks was the best PK predictor of response rate in the exposure response Emax model. This model predicted a CTRAF at 4 weeks value to provide 90% maximal effect of 128.5 microgram per milliliter, which is very close to the threshold concentration obtained in mice. For disease modeling, serum M protein data were best described by a tumor growth inhibition model, in which serum M protein is controlled by 2 processes: a growth rate and a cell killing rate, which is stimulated by izatuzumab exposure. This model provides a good prediction of the observed data and captures well different serum M protein profiles as shown in this figure. Using the established model, we evaluated alternative dosing regimen by simulating izatuzumab plasma concentration, response rate, and trends of serum M protein from baseline at week 8 and comparing with the original dosing regimen. The simulation showed that for weekly administration, together with a high dose of 10 or 20 mg/kg at the first cycle, allowed to quickly reach the efficacious concentration to overcome TMDD and to optimize the response. Higher response rate and greater decrease of tumor burden, his M protein, at week 8 were predicted for Q week for 4 weeks and Q2 weeks regimen compared to Q2 week regimen at 10 or 20 mg/kg. Therefore, this new dosing schedule Q week, Q2 week proposed by model and simulation was added after protocol amendment for evaluation in phase 2 monotherapy study and also implemented in the phase 1 combination study. The same modeling framework was applied to support dose selection in combination studies. Izatuzumab plus lenalidomide and dexamethasone RD or pomalidomide and dexamethasone PD. For izatuzumab monotherapy, 20 mg/kg Q week for 4 weeks, then Q2 week induced greater reduction in serum M protein. For izatuzumab in combination with either RD or PD, simulation yielded no substantial benefit in terms of M protein reduction when doubling izatuzumab dose from 10 to 20 mg/kg Q week for 4 weeks, then Q2 week. Altogether, izatuzumab 10 mg/kg Q week for 4 weeks, then Q2 week dosing regimen was selected for ICARIA pivotal phase 3 trial in combination with PD and other combination phase 3 study. Now I will present you the ICARIA-MM trial outcomes and the support of the most recent analysis in MIDD. As you can see here in the PFS Kaplan-Meier plot, addition of isatuximab to pomalidomide and dexamethasone was associated with significant and clinically meaningful PFS benefit in patients with relapsed refractory multiple myeloma. Population PK and exposure response analysis confirmed the selected dose and showed that a patient in the lowest exposure quartile still benefit from isatuximab treatment compared to the control group. With this outcome, we got FDA approval and positive CHMP opinion in March 2020. Then the question is how to use ICARIA-MM to build drug disease modeling platform to support dose confirmation and to support other clinical trial design. Joint modeling framework was proposed to characterize the association between biomarker kinetics, here is serum M protein, the characteristic of patient at baseline, and progression-free survival. The joint modeling analyzed the longitudinal data of serum M protein for both arms of ICARIA trial and its association to PFS. The tumor growth inhibition model in the previous steps were extended to consider the impact of the exposure of three compounds, isatuximab, pomalidomide, and dexamethasone in serum M protein killing rate. Of note, a new population PK model with time-varying linear clearance and nonlinear elimination was developed in the separate analysis and was used to predict isatuximab concentration for the joint model. The visual predictive trick showed that the joint model predicted well the observed M protein and PFS data in ICARIA trial. In the joint model, some baseline covariate was found to be significant. For example, albumin and beta microglobulin on serum M protein at baseline, IgG type on serum M protein growth rate, and presence of plasmacytoma on baseline hazard of PFS. Here, we evaluated if they have impact on serum M protein profiles and PFS curve. As you can see, patients with low albumin and high beta microglobulin tend to have faster rate of tumor regrowth at the end of the treatment, but similar PFS. Non-IgG patients tend to have faster tumor regrowth after 60 weeks, but similar PFS, even with twofold higher exposure at steady state compared to IgG patients. Patients with plasmacytoma tend to have much higher risk for progression. This analysis suggested no need for dose adjustment for subpopulation, in particular IgG patients, and allowed to confirm the approval of isatuximab dosing regimen, 10 mg/kg every week for 4 weeks, then every 2 weeks. In conclusion, model-based drug development has been successfully applied to support isatuximab dosing regimen selection in relapsed refractory multiple myeloma patients for combination therapy. Several analyses were performed to provide insight into dose and dosing regimen through preclinical to market approval and succeeded to support internal decision-making on dose selection. MIDD has shown to play a major impact on previous submission, for example, ICARIA, ICHEMA, and labeling for dose confirmation, no dose adjustment in subpopulation, and will continue to support future submissions and other phase 3 designs. MIDD increases both robustness of decision-making during the clinical development of isatuximab and the chance of success of the future phase 3 clinical trials. Two approval relative isatuximab 10 mg/kg every week for 4 weeks, then every 2 weeks, in combination with pomalidomide and dexamethasone, or in combination with carfilzomib and dexamethasone. We would like to thank CPATH for selecting our example for presentation at FDA MIDD training course. This presentation was sponsored by Sanofi. We also thank all the colleagues from different departments at Sanofi for their contributions, and thank you for your attention.